All right, welcome back. We're going to do the second half of the cell division lecture here. So, meiosis is the other type of cell division. Meiosis occurs only in organisms that use sexual reproduction. It is cell division that produces the gametes, right? The egg and sperm cells. Make sure you're aware of that word. If you remember from the last recorded lecture, the word diploid. Diploid means that something has two copies of each chromosome. So what does it mean if something has only one copy? Well, the word for that is haploid. And so what meiosis does is it starts with a cell that's diploid and ultimately produces 4 cells that are haploid. 4 egg or 4 sperm cells, depending on what sex we're talking about. So meiosis begins with one diploid cell, just like mitosis did, but ends up giving us 4 haploid cells in the end. And each of those haploid cells, one copy of each chromosome. This picture shows it quite beautifully. I'm not going to go through all of this, but I recommend that you look at it, maybe rewrite it if you want to get it ingrained into your brain. But I want you to notice for sure that there are 2 halves to meiosis. There's meiosis 1, which is going to be everything that's to the left of that dotted line. And there's meiosis 2, which is everything to the right of that dotted line. Meiosis 1 ultimately splits the original diploid cell into 2 haploid cells, and then meiosis 2 splits each haploid cell into 2 haploid cells. 2-part system. Meiosis 1 goes from diploid to haploid. Meiosis 2 keeps the number at haploid, but doubles the number of cells involved. I've used the word homologous once before, so I want you to be aware again of this term. Homologous means that chromosomes occur as a pair. If they're homologous, they code for similar traits, but they aren't necessarily identical. Again, on each chromosome you might have different versions of a particular gene. I believe the one I used was eye pigments. If you want, this might be a good time to go back and take a look at the previous lecture where I talked about the hypothetical chromosomes from mom and dad with the 2 different versions of the eye color gene on it. As a whole, all of your body cells are diploid, right? Your somatic cells, your brain, blood, skin, muscle cells, they're all diploid. But you don't want to pass on all of your genes to your offspring. I mean, in terms of biological fitness, living things do want to do that, but sexual reproduction doesn't work like that. You only pass on half of your chromosomes, 1 member of each pair. Just as you got 1 member of each pair from each of your biological parents. That makes sure that the number of chromosomes doesn't double every time there's a new generation. Meiosis. All right. Meiosis forms the gametes. Talked about that already. We talked about that already too. Okay, let's just get to it. So meiosis 1. Meiosis 1 starts with 1 diploid cell and ends with 2 haploid cells. Here is a hypothetical cell about to undergo meiosis 1. I want you to first of all notice something that's very different from mitosis. Notice that homologous pairs of chromosomes are associating very, very closely. Instead of having pairs of shoes randomly strewn about the nucleus, we've got each pair of shoes right next to the other member of that pair. That's very different from mitosis. So this is meiosis 1. This is the first part of meiosis 1. It's called prophase 1. The nucleus is still there, but starting to break apart. The mitotic spindle is starting to form, similar to what we saw in mitosis. Meiosis 1 has the same phase names as mitosis, except there's a 1 after it. So let's take a look a little bit more closely at what's going on here with the homologous pairs that are associating. So during prophase 1, homologous chromosomes closely associate with each other. This is prophase 1. During prophase 1, homologous chromosomes closely associate with each other. Remember that in mitosis, homologous chromosomes had nothing to do with each other. They're not just in the same vicinity during meiosis 1 and prophase 1. Homologous chromosomes are overlapping with each other and exchanging segments of DNA. The name of this process is called crossing over. If you remember, a gene is a segment of the chromosome that codes for a particular trait, and on any one chromosome you have potentially thousands of genes. Let's take a look at this example. We've got 2 chromosomes. Here's one of them. Here's the other one. And remember, each half of a chromosome is called a chromatid. So we've got 2 chromosomes, homologous chromosomes, and each chromosome has 2 chromatids. And so we have 1, 2, 3, 4 chromatids. Each chromatid, each one of these, will go to a separate egg or sperm cell. So each of these will become a different possible egg or sperm cell. Keep that in mind. Take a look at the different letters up here. We've got A, B, and C. Let's go ahead and break this down. Just so you know, eye color, hair color, freckles, it's not this simple, but I like the way that this works as an example. So the letter A represents the gene for your eye color. The letter B represents the gene for your hair color. The letter C represents the gene for freckles or not having freckles. Here are the dominant versions of these genes. Capital A is brown eyes, capital B is brown hair, capital C means that you have no freckles. Although I don't think that's actually dominant, but you get the idea. Lowercase a is the recessive gene for blue eyes, lowercase b the gene for blonde hair, and lowercase c the gene for freckles. So we're going to say that this person got the red chromosome from their mom and the blue one from their dad, because you get one member of each chromosome pair from each parent. The chromosome from their dad has the gene for brown eyes, brown hair, no freckles, because that's what their dad looks like. The chromosome they got from their mom, red one, has the genes for blue eyes, blonde hair, and freckles. Keep in mind, since they're homologous chromosomes, they have the genes in the same spot to code for the same stuff. You're just going to have different versions of it. Crossing over is when homologous chromosomes exchange segments of DNA. Some of this exchange has happened here. We've swapped maternal and paternal chromosome segments. What does that do? Well, it ends up producing combinations that we didn't see in this individual's actual genome. There's the 4 possible gametes you can make. This one, we're going to say it's a female, so we'll say they're egg cells. This is one possible egg cell that could be made for this chromosome, with this chromosome in it. And I don't have what the letters stand for here, so let's just go back and look at it here. Here's one possible chromatid, aka one possible egg cell. If this gets passed on, the child will have the genes for blue eyes, blonde hair, and freckles from this hypothetical mother. If this gets passed on, this egg cell gets fertilized, the child would have the genes for blue eyes, but brown hair and freckles, or brown hair and no freckles for here. If this one ends up becoming the egg that gets fertilized, brown eyes, blonde hair, freckles. This one is the egg, becomes the egg that gets fertilized, brown eyes, brown hair, no freckles. We've got a new combination of genes that did not exist before. That's the whole point of crossing over. So each chromatid will become a separate gamete, thus crossing over produces gametes with a new combination of genes and chromosomes that include snippets from both paternal and the maternal side. So that was all happening during prophase 1. Metaphase 1 sees the chromosomes aligning at the equator of the cell, just like in mitosis, except note that now they don't occur in a single file line, they occur in pairs. During metaphase 1, homologous chromosomes align in pairs at the equator of the cell, with one member of the pair above the imaginary equator, which we're going to draw right there, and the other chromosome below the equator. So each pair aligns together with the other member of the pair. What determines which chromosome is above and which one is below the equator? Sheer random chance. And each pair aligns independently from the others. So this first pair, random chance has determined that the blue chromosome, we'll say that's from the father's side, is above the equator. Random chance has determined here that the blue chromosome is also above, but here it's below. And if this is a human cell, you would have 20 additional pairs that would each randomly assort themselves, and sheer random chance determines which goes which. If you can visualize what's going to happen, the cell's ultimately going to split like this. And so that random assortment means that you end up with combinations of chromosomes that include both your mother's side and your father's side. So your mother's side and your father's side. And so the law of independent assortment says each chromosome pair randomly aligns itself during the formation here of meiosis 1, metaphase 1. And you get a mix of both paternal and maternal genes ultimately on either side of that line. I want you to kind of visualize how they're splitting. In anaphase 1, what happens is the chromosomes split, and I've tried to show a little bit of crossing over here by including snippets of DNA segments having been swapped. But notice how each member of a homologous pair is being plucked apart from the other member. If you remember back to meiosis, or mitosis, we separated the chromatids. We pulled them apart. In meiosis, the chromatids stay together, the chromosomes are separated. In anaphase 1, homologous chromosomes separate from each other, moving toward their respective north and south pole centrosomes. The law, this is the law of segregation. It's one of Mendel's statements that he came up with, is that during gamete formation, which is what's happening here, homologous pairs align and then separate during anaphase 1. Mendel didn't know about anaphase, but it's basically so that you end up with daughter cells that are going to have this, not have two copies of the same chromosome. Because you also then are going to have this hypothetical egg cell get fertilized, which would add an extra copy of the chromosome if the law of segregation didn't work. What happens if it doesn't work? You end up with what's called non-disjunction. This picture shows that failure of chromosomes to separate. So here we have a chromosome that is clinging to the other member of the pair, and so both of them get inherited by this one daughter cell, which ends up producing gametes that have two copies of this chromosome, and gametes that are missing the chromosome over here. Non-disjunction results in individuals receiving an additional copy of a chromosome or missing one. Most chromosomal non-disjunction abnormalities don't even confer survival to birth, but for some of the smaller chromosomes with fewer genes, non-disjunction still allows for survival and even thriving. Down syndrome, for instance. Trisomy 21 is the official name for Down syndrome. The 21st chromosome is among the shortest of the chromosomes that we have. This individual has a karyotype here. With Down syndrome, they had an additional copy of the 21st chromosome. A couple other non-disjunction examples that still confer long-term survival involve non-disjunction of the sex chromosomes. Now, if you are a female, you are XX, and if you are a male, you're XY. But sometimes there are errors in the copying process of those. Non-disjunction occurs, and you might get an extra X, extra Y, maybe missing an X. So what are some possible things that can result from that? One of them is Klinefelter. That's XXY. A male with Klinefelter syndrome would still have sperm production and testes, but you might have poor muscle development, secondary sexual characteristics, maybe breast development, for instance, because of that additional X chromosome. Turner syndrome occurs in females with just an X. Generally, women with Turner syndrome look a lot younger than they are. They're shorter in stature and often are infertile. If you watch Law and Order, NCIS Los Angeles, I forget, one of the actresses in there has Turner syndrome. I forget her name. Poly-X syndrome occurs in females with an extra X. In terms of observable differences, there are none between poly-X females and regular females. So if you are watching this and you're female, you could be poly-X. You wouldn't know unless you had a karyotype done. Some males will be poly-Y. There was a debunked study that showed men in prison tended to have higher numbers of this abnormality, but that is not the case. But those are some examples of non-disjunction and the effects that they can have. Again, typically if you have 3 copies of some of these larger chromosomes, that doesn't even allow for survival to birth. So now we're in telophase 1. In telophase 1, we have cytokinesis, which is the cytoplasm splitting. And by the time this is done, we have 2 daughter cells. 2 daughter cells, each with 1 copy of each chromosome. Again, notice that the sister chromatids are still attached to each other. They might not be identical anymore because they've got crossing over, which has resulted in some of the swapping of DNA among homologous chromosomes, but they're still attached to each other. And so we've got 2 new daughter cells, and they're going to be haploid. 1 copy of each chromosome instead of 2. Meiosis II is nice and simple in the sense that it is identical to mitosis. The only difference is our starting point. In mitosis, we start off with a cell that has 46 chromosomes, and we end up with 2 new daughter cells that also have 46. In Meiosis II, we've already done Meiosis I, which gave us the haploid 23 chromosome daughter cell. In fact, it gave us 2 of them. And in Meiosis II, we're just going to do the same process as mitosis, only we're starting with 23 instead of 46. I think I have it illustrated on the board. Yeah. Prophase 2, metaphase 2, anaphase 2, telophase 2. Welcome you to pause this, take a closer look, but you've got the same stuff happening as we saw in mitosis. The only difference is we're starting with half of what we had compared to mitosis. By the time we're done, we've got 1, 2, 3, 4 daughter cells, and all of them are haploid. These will become egg or sperm cells. If you are female, then the number that are produced during a lifetime in terms of eggs will be in the thousands. If you're male, this will happen in the hundreds of billions. That's how many sperm cells the average man makes during a lifetime. Last thing on this PowerPoint is polyploidy. Basically, polyploidy involves non-disjunction not actually producing haploid gametes. In humans, this would look something like this. 46 is supposed to get to 23. It doesn't. It stays at 46. These end up becoming sperm cells. They fertilize an egg cell that had the same issue. And voila, we've produced a human zygote with 92 chromosomes. 4 copies of each. That's polyploidy. Here's a visual. 3 pairs of chromosomes. This is what's supposed to happen. However, if this does not happen, and instead 2 gametes get made like this, we end up with an individual with 4 copies of each chromosome. You might expect that that's bad, but it's not always the case. Many different varieties of wheat are different because they have different numbers of chromosomes. Some are diploid, some are what's called triploid, 3 copies of each, some are tetraploid, and some are even more. One example of an animal species that may have arisen from polyploidy is the gray tree frog, which can actually be green, hence the picture here. There are 2 species that look almost identical. Actually, they look identical. 2 species of tree frog here in Missouri. The only difference between them is they call a little bit different, and one of them has exactly twice as many chromosomes as the other. So they think that sometime in the past, there was a copying error, and a small population of these tree frogs ended up with twice as many chromosomes as the other one, and didn't really seem to affect them negatively. Natural selection favored them, and what do we have? A new species of tree frog. So speciation can occur due to copying errors of chromosomes. That should be the last slide for this PowerPoint. Very good.